Chunhua got her Ph.D. in Biomedical Informatics at the University of Washington in Seattle, and then did a fellowship in informatics at University of Pittsburgh, and then came to Columbia University in 2007 when I was there. So we overlapped by about a year, and started publishing not too long after, co-publishing not too long after that, with 15 publications together. She has too many things to enumerate, but to give you an idea, publishing over the last 22 years, she has 376 publications worth almost 10,000 citations. H index of 46, and I-10 index of 173, and so she's also been honored by fellowship in the College of Medical Informatics, which she has her pin on, and she's also a fellow of the International Academy of Health Sciences Informatics. And she is nationally known for her work in clinical research informatics, especially with phenotype detection, and is a major contributor to national consortium, emerging consortium, and CTSA and others. So very happy to have you here and hear about what you're doing with clinical research informatics.
Thank you so much, Jim. Hi, good morning, everyone. Thank you so much for having me. It's my great pleasure to be here today. So Jim has been very supportive and has been providing strong mentorship to me since I joined this field. As Jim mentioned, I'm a clinical research informatics researcher. All my research has been surrounded using informatics methods to make clinical research more efficient, make clinical research results more generalizable. I was trained as a computer scientist, so the early methods or early work will really focus on expert system, knowledge representation, and ontology development. And later we are using large-scale clinical data to support knowledge acquisition and knowledge engineering. And also, we also focus on the user requirement and user needs, and using the current hot buzzword, which is human-centered AI, and also looking at the bias and trustworthiness of AI methods and applications. So the ultimate goal of clinical research is really to benefit human health. So I'm hoping in this talk, I can kind of review the challenges in the current evidence-based medicine, and also propose the concept or conceptual framework called next generation evidence-based medicine, and then kind of sample some research and applications that using informatics approach, informatics interventions to reach that goal. So evidence-based medicine is a paradigm introducing like 1990s, emphasizing the systematic approach to medicine where doctors and healthcare professionals can use best available scientific evidence from clinical research to facilitate medical decision making. I'm sure many of you have seen this diagram. This is from a report from National Academy of Medicine, talking about the problematic evidence pipeline, which starting from the science, much of the science didn't lead to robust, reliable, reproducible, or replicable evidence. And then, even though some evidence gets generated, and only a small portion gets translated into a care setting to benefit human health. So a lot of experts calling our evidence-based our current clinical evidence pipeline is kind of full of missed opportunities and waste and harm to patients. And so just to mention some example problems in our current evidence, and in recent years, the lack of diversity in clinical research has been called out by many field leaders and experts. So these are some like top journal articles, publications talking about the harm and significance of lack of diversity. And particularly, not only in general medicine, but also in genomic medicine, the lack of diversity can also be very harmful. That affects the frequent reclassification of genetic mutations or variants, and that can potentially may cause or lead to unnecessary surgeries for patients once they find out they have a pathogenic variation, but then later publication can show that variation is non-significant. And then, the whole world experienced the COVID-19 pandemic, and the problems in our evidence pipeline, it kind of exacerbated during that difficult time. A lot of underpowered studies get published quickly, and then so the literature is growing exponentially in a short time, but then the meta-analysis didn't catch up. The information overload, the difficulty and time consuming to generate guideline and meta-analysis. All these other problems become very obvious, and especially during the pandemic. And then for simple questions such as which treatment can improve any disease outcome, and using COVID-19 as an example, you can frequently see top journals publishing contradicting evidence. And this is articles published in a very short time period. They had different conclusions regarding the effectiveness of this drug, TCG, treating COVID. They use different outcomes, they use different study populations, and so on. And as I mentioned, in the genomic medicine, the prevalence variant reclassification also affect cancer genetic testing, and this is very frequent. Some cancer patients, they may experience surgeries, but then later find out, variance is not really significant. So in addition to the lack of quality and then the lack of robustness of evidence, another challenge is in implementing evidence, which is another phase in evidence-based medicine. This is due to the information overload. As this article mentioned, evidence-based medicine really emphasizes the skills for doctors for efficient literature searching and application of formal rules for evidence appraisal. But this is hard, right? Given there are so many trials and meta-analysis published very quickly, and no human beings can catch up with the growth of the literature. So a few years ago, my postdoc looked at the evidence appraisal, the current status, and we identified many areas are not leveraging the current advances in technology. So we don't have infrastructure and data standards for data acquisition, information management. As shown in this diagram, those blocks captured by the dotted or dashed lines, they are severely lacking or almost not there, rarely exist in the current evidence appraisal pipeline. So the conventional methods for evidence appraisal is journal club, and however, the discussions may not be captured systematically or saved as records that can be for later reuse. So in that study, we really identified the need, which is unmet for evidence appraisal. This is also like where informatics can potentially help. So let's go back to this picture about the problematic evidence-based medicine. I think in this field, people are really emphasizing, how can we make science more robust, leads to more quality evidence, make the evidence generation process more efficient. And then the next phase is make this evidence accessible, available at the point of care. So here I want to propose the concept of the next generation evidence-based medicine, which is minimize the waste and improve the quality and reliability at phase 1, which is evidence generation, indicated by the number 1 icon, and then followed by the evidence implementation at the point of care, which is number 2. But I also want to add 2 more phases, which is phase 3 and 4. Phase 3 is really to broaden our view of evidence. So enable a learning health system by incorporating more diverse data resources and methods for evidence generation to support continuous learning and update of evidence, because evidence is dynamic. And then the next one is to really use the evidence to inform more robust science, and I call it evidence-based research. So let me go into the details of this new conceptual framework. So this framework really emphasizing closing the loop, right, from evidence generation to evidence implementation, and then using evidence to enable the more precise research, called evidence-based research. So Jim has published an inspirational paper called Desiderata for terminology design for clinical informatics. And here I'm just borrowing Jim's idea about, what's our desiderata for each phase of evidence-based medicine. So I think for evidence generation phase, we really want to achieve more efficient process and a more robust and high quality, non-biased evidence and better generalizability of the findings to different populations, different settings, and so on. And then in this phase, the key problems are insufficient delayed recruitment and lack of diversity in study population, lack of representativeness, and lack of generalizability. And then the second phase, which is evidence implementation, the desiderata is the scalability and also the make technical language in scientific publications more comprehensible, computable for decision support. So the big challenges in this space is really the information overload and also implementation chasm that has been talked about, published on by many informatics leaders. Particularly, even we have clinical practice guideline, but to implement in the EHR system, that's difficult. And information overload, I already mentioned, given so many papers published, how can we scale the meta-analysis? And then the phase 3 is evidence refinement, support continuous learning of evidence, so that the desiderata is a timely, rapid learning, enable a systematic approach for evidence appraisal, make the appraisal information or knowledge broadly shareable, and enable a learning system or infrastructure. And then the last phase, which is new, called evidence-based research, really emphasizes a systematic approach to systemize the process. And so the future research can be more rational and fair and patient-centered. Make sure the research using the relevant representative patient population and incorporate the patient-centered outcome, and then addressing the urgent evidence gap rather than just repeating what's already done for decades or many years. So our lab is very fortunate. Over these years, we have been receiving research support to tackle the challenges in each of these phases. So for example, for evidence generation, my work has been focused on developing informatics interventions to make eligibility criteria computable in a hope that we can enable or empower clinical research coordinators to better translate criteria into cohort identification queries in a natural language user interface. And then for evidence implementation or adoption, the phase 2, we were funded to develop information evidence extraction and summarization pipeline. And then we are also funded by eMERGE, the UO1 mechanism, to look at the implementation barrier, what model is portable to enable the integration of research as part of the patient care process. For phase 3, we have a grant from NHGRI looking at how can we develop fair phenotype representation to enable continuous variant reclassification using the rich phenotype from EHR data to really show the learning health system for evidence related to genetic variants and using the rich clinical data. And then for phase 4, evidence-based research, we leverage the existing funding resource to look at how we can use existing real-world evidence to inform the clinical trial design, particularly to optimize eligibility criteria optimization, make sure the criteria, the study population are representative of the real-world population, and also to identify evidence gap systematically. So to allow the future clinical investigators and researchers to be better informed about which areas needs more evidence and should be prioritized when they design the next study protocol. So, here, for the next generation evidence-based medicine, I also want to point out we need to have a broader view about what is evidence, right? So here, this is a hierarchy of scientific evidence. I have seen it since I was a student. Really emphasize, randomized controlled trial, the gold standard for generating evidence, followed by meta-analysis. But I think nowadays we should not be limited by RCT, right? RCTs can be slow, take years, and then you have to wait until RCT finished. Many RCT finished, then meta-analysis generate clinical practice guideline. So I think the emerging of the real-world evidence, many rich multi-level patient data, all these should be kind of broaden the opportunities for other methods for rapid evidence generation. So I think for the next generation evidence-based medicine, we should also incorporate this into our view. Sorry, the title is the comparison between current evidence-based medicine and next generation evidence. So I think the methods, as I mentioned, need to be more diverse, not limited to RCT. And then the timing for evidence adoption, I think we should not wait until the formal guideline, which we can consider real-time decision support using the real-world evidence. And for evidence appraisal, we should leverage the modern informatics data science network technology to enable data-driven and stakeholder-centered evidence appraisal, not be limited by the journal club or social media. And for evidence synthesis and dissemination, we also have fantastic opportunities enabled by the large language model, and hopefully that can improve the efficiency and scalability. And then, as I mentioned, we also identified examples of using real-world evidence to inform future research and to use large language models for high-throughput evidence computing, including evidence knowledge representation, building structured evidence base, as well as incorporating the future patient opinion values into the evidence-based practice. So these are the kind of the differences we identified between the two versions of evidence-based medicine. So next, let's look kind of a big overview of what informatics can do for evidence-based medicine. So next, I'm going to just quickly sample some projects I mentioned in these 4 phases. So phase 1 is to use informatics to improve the efficiency for evidence generation. And then, as I mentioned, recruitment is the #1 barrier for clinical research. So for many years, I've been focused on this topic because my collaborator and mentor is a clinical trialist, and then he told me, this is the most significant barrier for clinical research, and where it's also a place where informatics can bring the best value. So this is a system we developed called criteria to query. And what you see is one older version of the user interface. You can enter the clinical trial NCT ID available from clinicaltrials.gov. Then the system can automatically crawl studies eligibility criteria text, or you can copy and paste eligibility criteria text from an existing clinical trial protocol. And then the system can parse this criteria, recognize the concepts in the eligibility criteria can be diagnosis, measurement, medication. So here indicated by different colors. Each color represents a semantic type. And then the system then translates this into the executable OMOP common data model standards-based cohort identification query that you can share with anyone with a database of patients and to tell you, how many patients satisfying these criteria. So this system has gone through 3 iterations. The first version is purely automated, and we just dump in a free text, get a query. And but then we learned that's not good enough because in reality, no coordinators just dump the entire protocol's criteria into a system because some criteria are not queryable. So then it evolved into the second version, which is developed by my current PhD student, Ilu. She incorporated some human intervention into the query formulation process. So after NLP extract and recognize all those concepts and relationship, and she designed a user interface, interactive user interface to allow users, coordinators, to specify which criteria should be returned for a database query and which criteria should be refined. So here we call it human-computer collaboration. This is also the concept of human-centered AI. Allow the human and computer kind of divide and conquer. So we identify the following tasks that can be automated to leverage machine efficiency, such as entity recognition, concept recognition, and concept normalization, and establish the initial mapping to the standard terminologies such as OMOP common data model in this example. And then dependency relationships, extraction, like which value attribute or threshold should be attached to, say, lab test concept. And logic extraction, phenotyping, and you can link your phenotyping database, algorithm database to the concept. So for example, if you're looking for diabetes patients, you can recognize the concept diabetes, but what does it mean to have diabetes? You can map it to the algorithm library that's already available. And then query formulation. So these can be automated by the machine, but it's still not enough. So we first let our pipeline to do all this, but then we let the human to intervene. So the human can add their selection about which data source should be plugging with the query. Like because for EHR data space or information space, a lot of information they are redundant, but certain information source is more reliable, higher data quality than others. And then another example is the criteria prioritization. So the eligibility screening we learned is not one-step process. Coordinators tend to divide the criteria screening into multiple phases. They will first start with major eligibility criteria. You try to rule out a lot of definitely ineligible patients, and then they will have a much smaller pool to go through the lengthy, more detailed criteria. So we allow users, if it's like initial phase, just for feasibility analysis, they can just pick certain major criteria. They don't have to use the entire protocol, for example. And then concept granularity, the machine picked the concept mapping results may not be perfect. It depends on the context, it depends on the clinicians or researchers' preference. So these are the tasks we allow users to intervene and add their opinion before the query is generated. And that's like our workflow to finish the query. Start with machine, which is indicated by these orange lines, and then indicated by the blue box down there. And then we allow users to add orange input indicated by the orange lines on the top. and then we eventually will generate clarity. So that's the step one, which is the evidence generation process. How can we facilitate coordinators to take the criteria into more efficient process for cohort identification? So next, I'm going to move on the second phase, which is evidence computing. How can we make the evidence available in a PubMed as free text, make that more available for implementation? So what we have been doing here, this is a paper entitled Evidence Map. So we take the RCT publications in PubMed, and we start with the abstracts and title, and we extract all these medical evidence entity represented by the PICO framework. As you know, a lot of clinicians using PICO to query the medical evidence. So PICO stands for Participant Intervention Comparison and Outcome. So we build the NLP pipeline to extract all the PICO entities. Then we construct the medical evidence proposition, linking all these entities with each other. So then the next level is the evidence map. Like once you can build the proposition, then you can kind of group them into different arms that represent entire structure of evidence for the entire study. So here are some examples, like free text, 7% alcohol is more effective in reducing number of CFU centimeters after 2 minutes. So the 70% alcohol is labeled as intervention, and then the observational the relationship is between this intervention and outcome of number of CFU. That's it's more effective in reducing. That's our observation. So we form these propositions in our output, and then once we have this. And this proposition is based on the medical evidence entity dependency parsing. So we have a dependency parser in our pipeline. And then once we have this, and we can use this medical evidence map representation tool, work together with the attention model to detect which sentence of the abstract contain a finding of this article. So we build these neural methods to automate the extraction of findings from the PubMed abstract. So I will be quick here. So that was a few years ago, and then recently, given the quick rise of the large language model and ChatGPT, we're kind of starting to experiment with GPT technology for evidence extraction, retrieval, and then summarization. So this is a recent article we published. It's a perspective. So we assess the potential role of using generative AI for evidence retrieval, synthesis, reasoning, and dissemination. So we identify these use cases, and this is also kind of ongoing research that we are exploring the effectiveness of using large language models for these purposes. And we also did some experiment to evaluate the large language models on medical evidence summarization. The most critical need for this purpose is really to evaluate or establish novel evaluation metrics. The traditional precision recall may not be sufficient. So right now we need to consider, the readability and other metrics that's more relevant to all these machine-generated summaries. And here we use these metrics to compare these different models of ChatGPT and to assess the summarization performance in this automated evaluation. So I won't go into the details there. Sorry. And then next, another technology we have been using is this retrieval augmented generation. It's called RAG. So we have been using that to facilitate scientific claim verification, and this is also a recent study. people can enter a claim, and then the system has 3 steps. First, to retrieve relevant abstracts for this claim, and then we automatically identify the sentences that contain the findings relevant to this claim in each article, and then we perform a label prediction. Is this a finding supporting or refuting the claim? And then stratify or group the abstracts into different groups, like supporting the claim, neutral, or refuting the claim. So it's very simple classification system, but leveraging the latest RAG technology. So now moving on to the next phase. So I talked about, evidence generation, evidence summarization, evidence retrieval. Next is really how can we acquire new evidence from large scale real world data. And so this work started maybe 7 or 8 years ago. We recognize, for phenotyping, we still have a lot of unknowns about, disease, what are the characteristics of different diseases. And then patient records, especially clinical notes, contains a lot of nuanced phenotype knowledge. So that's kind of started our project. So we developed a system called EHR Phenotyzer, take all the EHR notes, extract the phenotype concepts, and then perform phenotype concept normalization, and build, new knowledge base of phenotypes. And then we use this phenotype to feed into a knowledge engine, contains all the terminology such as OMI, HPO, human phenotype ontology. OMI stands for online Mendelian inheritance in men, gene ontology, and so on. That was led by my collaborator, who's a bioinformatics expert out of Taiwan in CHOP. And then we feed all these EHR extracted phenotype concepts into that system called a Phenolizer, and we expanded into EHR Phenolizer. And then once we go through that knowledge inference engine, then we can get ranked a list of ranked genes as possible genetic or genetic underpinning for the disease or conditions the patient may have. Then the patient goes through the clinical exome sequencing, looking at the variants of the sequencing results, putting these together, then we will see possible diagnosis. So here are the example of what we can do with the clinical notes. We can first extract the phenotype, map them UMLS, and then later to human phenotype ontology. And then once this concept gets normalized, goes through the Phenolizer, we get a ranked gene list. And then we combine this ranked gene list with the clinical exome sequencing results. Then we can kind of see which genes show it's shown in both lists, and then identify potential causal genes for the patient. So that's how it works. And then recently, we are also leveraging large language models to replace the conventional methods for concept extraction and normalization. So this is an example of fine-tuning large language models for rare disease concept normalization. So we did an experiment to compare, the general purpose large language model, is that good enough? Or, how is that compared to dictionary lookup approach? And then we also looked at how fine-tuning means using a domain-specific data sets to train a model. And then we showed that our results showed the fine-tuned model actually achieved better performance than dictionary lookup approach, and also better than general purpose large language model. So here you can see if you have a dictionary lookup approach, if you have any typo, you possibly will miss identifying that concept. But then if you use the typical NLP or conventional NLP algorithm, if you start with the text like expanded blood vessel, and then it may be misidentified. But we use the fine-tuned model, we can still recognize, okay, this text or this term is associated with the HPO concept, vascular dilatation. So we are using this to automate the concept mapping process. And then, so our early results, this is not being published, but these are our early results, shows the fine tuning is effective and it's the right way to go. Another thing we have been doing, this is another experiment by a different student looking at, if we want to extract a phenotype, which method is more effective? Should we just extract using a free text as input and then go through the HPO concept extraction and then use that extracted HPO concepts to drive the diagnosis of rare disease? That's one option. And the other option is just feed the free text to the large language model, just use the entire narrative as input and then use that directly to generate diagnosis recommendations. So we compared these two options. Our research questions really ask, is information extraction still necessary and useful when we are using large language models? And then for this experiment, which our key finding is that information extraction, like going through the step of extracting human phenotype concept, human phenotype ontology concept is still very necessary. It's actually better than just throwing a chunk of text into the GPT. So that's our preliminary finding. And then so we have this methods to scale the knowledge acquisition, information extraction, real world evidence extraction from large scale real world data from EHR. And so the next step is really kind of use this to generate large real world evidence. So this is an example of using our entire Columbia University Medical Center's clinical notes to generate knowledge about rare disease using our previously mentioned NLP pipelines. And we built this large scale data sets called open annotation for rare disease data sets. And the technical details is in the citation. And so this is a completely data driven involved, and we apply it and test the external validity in both in TRAP data sets as well. We started with Columbia, but we incorporated two medical institutes data. And more than 10 million of individuals, we use both structured and unstructured for diagnosis. And we also so in this work, we actually identified more than 2 million novel disease phenotype associations of pairs. And we performed some manual review, achieved a precision higher than 60%. So these are the detailed steps involved in our methodology pipeline. We leverage IAHSI, and then we also use the human phenotype ontology, and then we apply some statistical descriptors to calculate the odds ratio for the pairs of genotype disease association. So I will show you how this can be useful for that rare disease diagnosis. So one way is people can enter, we call it phenotype-driven rare disease diagnosis. Like a user, we have a web user interface for this use case. People can play with it. It's publicly available. So one way people can enter a list of phenotype concepts as shown on the top left, and then you can specify the age group of the patients. And you can also specify how many, top concept you want to return. You can return top 10, top 20, or top 50. And then the results is displayed at the bottom. You can identify the diseases that contain or this entire set of phenotypes ranked by their odds ratio. And the other way is you don't start with phenotype, you start with disease instead. And you enter the disease, human phenotype ontology concept, and then you specify, how many top associated phenotypes you want to return. And then you click the submit button, you will see a list of phenotypes associated with this disease. So here we separate them into known associations as well as novel associations retrieved from our real world evidence. So here for this example disease, the 3-1 indicated by the green arrow, so it's already known, it's already in the literature, but we also identify some previously not annotated associations. So this could be a potential new evidence that still needs to be verified in future studies. But at least this is a way to use large-scale real-world data and real-world evidence to inform the following research about phenotypes to better characterize rare diseases. And then, so that's an example for genetic research. And then another example of using the real-world evidence to inform research is really to improve the precision of research design. So this is a book, it's a blurry picture, apologies for that. It's from a textbook for clinical trial design. And all we get is, this looks like it's for designing a hypertension trial. And then they looked at the distribution of systolic blood pressure of hypertension patients. And then the trial designers will draw 2 lines, vertical dotted lines, as you see here. They will decide, okay, to the left of the first vertical line, there are people too healthy to be treated. And then to the right of the second vertical dotted line, there are patients too sick to be treated. So when we design a trial, we got to pick the people between these 2 lines. But the question is, where should you draw the line, right? And then will you unnecessarily exclude people that should benefit from this research? And how do you make rational decisions regarding the threshold? So that's the old school methods for designing eligibility criteria. We also have other publications talking about similarities between a lot of the protocols in the same clinical domain, like diabetes trials tend to reuse a lot of eligibility criteria, even the same threshold for HbA1c and other quantitative variables. So what we were proposing is a system or approach that leveraging real-world data to inform eligibility criteria design. Let's start with real-world data and phenotyping results, and then to identify patient subgroups with different characteristics and different outcomes. And so that's the data-centric path. That's not enough. We also need a knowledge base, which is on the top. We look at all the existing finished trials, always have public data, publicly available knowledge, and then we identify commonly used eligibility criteria, and then look at their recruitment outcome, frequency chosen thresholds, and then matching this knowledge with the real world data. And we propose this should be the future way designing eligibility criteria. And then we should bring this information to whoever has a role in selecting the eligibility criteria, to be clinicians, researchers, and in the future even including patients, to let them make informed decision about what eligibility criteria should be for future to be funded study. And then we also incorporate operations research, existing well-established methods called multiple attribute decision making for inclusion criteria optimization. And then this model was just recently published. We emphasize optimization for safety, quantity, and feasibility, which is quantity, number of patients will be eligible. Safety will be measured by, if you set this criteria, how many patients will experience adverse events based on their EHR records, and also the diversity. Diversity can be measured by, diversity in age, gender, race, and ethnicity. So, So that's another way to use real world patient data, real world evidence to inform the study design and the clinical research design. And we also tested in different disease domains. I think we have 2 papers. One is cardiovascular disease, and this one is in using the real COVID-19 patients to inform the appropriateness of COVID-19 clinical trials. Which one criteria is more restrictive and exclusive, which criteria can minimize the cohort very quickly. So that's phase 3, and then phase 4. We also use this to identify the evidence gap. So this is a study published a couple years ago. We looked at, from clinical trials to clinical practice, how long are drugs tested in the trial phase, and how long are drugs used in the real clinical care setting. And we compared the length of we looked at the length of clinical trial for many drugs, and we also looked at the length of use by patients taking these drugs using a large scale EHR data. And we do identify a lot of the drugs, a significant portion of drugs, they were tested in a short time period, but once they hit market, being given to the patients, they will be being used by a lot of patients for much longer, time period. So then the question is, are they safe for patients to, be on such drugs for much longer time period? So this is unknown. For us as an implementation, our job is just to make all this transparent, and then to point out, these can be potential evidence gaps, and we can do this using all this public data and patient large scale real world data. So I was rushing, sorry about that. I think the key takeaway is, evidence-based medicine moving forward is really facing a great opportunities given the advances in biomedical informatics, data standards, big data, and also generative AI. So I think there are a lot of things we can do for, better process for evidence generation, better efficiency for evidence extraction, retrieval, and summarization, and also how we can use diverse real-world data to better inform clinical research design, to better prioritize research that will address urgent evidence gaps. So I think these are I showed you some examples that things we can do, but I would say this is really the beginning of the process, because I think more needs to be done. And another key takeaway I want to emphasize is big data. We already see many people talking about big data, but I think now, given the rise of generative AI, knowledge is far more important than data. We'll make sure this AI is reliable and trustworthy, because as our experiment shows, if you don't let the AI work together with the existing knowledge, the AI can make mistakes. They can give you a fake concept ID from that doesn't exist in human phenotype ontology. So fine-tuning, retrieval, augmented generation, all these technologies that incorporating knowledge and into the AI model is very important. And another thing is the human-centered, put human in a loop. That's another methods that we should always leverage. In future work, there are many for next generation evidence-based medicine. I think I don't have time to cover that, but we all know social ethical issues. I think there's still a lot of controversy regarding real world evidence. Is that good? Should that be, recommended or allowed for real time decision making? So I think much more remains to be done in that area. And when we incorporate patient reported for evidence outcomes and for evidence appraisal, for evidence generation, I think we need more work there. And finally, implementation of evidence at point of care is still difficult. I think many of our field leaders, such as Samson Tool, Ida Sim, Mark Newson, spend lots of time, major part of their career developing computerized clinical practice guidelines, including TED-Shortlift. But it's hard, right? So I think we still need to think about how can we implement all this evidence at the point of care. What needs, what mechanism will make this evidence computable, accessible to a lot of clinicians, providers out there. Still, many research questions remain to be addressed. So I think I will conclude it here. Oh, I want to say, I feel most of the time is my research, my students and trainees kind of push me forward. All these works were done by them. All these models and algorithms, implementations. So these are my lab members, and I also have a lot of collaborators, including Jen, Patrick Ryan, Wendy Chang, many. So I want to thank them. And finally, I want to thank you for your attention. Sorry for a rush. Thank you.
And thanks for the great presentation, covering a lot of different things. And we do have a question from the audience. Dr. Reddy, I don't know if you can unmute yourself, or I can also read your question. You put it in the chat.
So Amy, thank you. I suggest me look at chat.
Okay. Although it's generally accepted that EBD is the standard of care, I do see hospital tools that use tools which have not been vetted. Actually, no, sorry. The question before that. EHRs are institution-specific, not publicly available. How can large language modeling work with evidence-based research?
Yeah, thank you. That's a great question. We all know that actually our experiment with large language models, so far, we are still limiting the use with the publicly available EHR data and publicly available PubMed data resource. So we have not used the large language model with our own notes. I have to acknowledge that because we don't get the approval for our enterprise to use large language model. So I talked to several of you yesterday that that's still pending. I totally agree because the reliability and safety and patient privacy, these are addressed or unsolved problem. But I think I'm hopeful this can be resolved in the future because that's the trend. I think no one can avoid that.
Yeah, I also know from other institutions that they will actually run the language model inside their firewall and on their own data, so that way yes, everything's protected. But that doesn't contribute to the general scientific knowledge. And the other question, another question.
Well, I have a quick question. I was saying that.
Yeah, we're gonna go. Let's have questions from all the folks that actually showed up.
Oh, yeah, you know what? I can't see them, so I'm going to let you MC, and then maybe we'll go back later to some people on the virtual questions.
Okay.
Great work, great talk. And I have a big question regarding the phenotype and the rare disease project. And where are the mutations that you collected publicly or for the rare disease? How do you collect the genetic information?
Oh, so for these patients, so we have a set of patients already have the sequencing data. And then we also have the electronic health records data. So when we test all these methods, we always use this cohort. Then we can kind of extract their phenotype. We can see how the phenotype-driven approach can arrive at the list of prioritized genes. Then we go to their sequencing data and see which variants of genes is shown in their sequencing.
How many samples did you use for this project?
How many samples? Yeah, so my collaborator for this project is Dr. Kai Wang in CHOP. So he has collaborators in CHOP, has many patients, children, primarily children. And then another collaborator is Wendy Chang. So she also has a I don't know the exact number, but at least more than 10,000. Yeah, so we used it. Yeah.
And a follow-up for this one, and how many of them are, you mentioned there's some novel discoveries based on the phenotype. So how many of the variants of the disease can be explained by the novel discovery? What's the percentage you have here?
You mean what percentage of the derived phenotype and the disease pairs are considered novel?
Yeah.
Yeah, it's actually a large number in that paper. So in that paper,
Yeah, I think that's significant because like for even for rare disease, right? Less than 30% can be explainable. And there's like many undiagnosed disease, right? In terms of rare disease. I think that's very, very helpful.
yeah, but the future research still needs to be done to verify this, because this is kind of showed us possibility of using large scale real world data to generate new evidence that's not known from previous literature. Yes, I think here, this is from my students, he identified more than 2 million novel disease phenotype pairs. Yeah, and I think recently article from all of us, all of us is a national cohort. They also said they have 275 million, novel variants. So that's a recent NIH director news announcement. So clearly we can see this real world data have a lot of potential.
Thank you.
Thank
I
you.
have to go.
Yeah, Jim.
So the last study that you showed, the gap between
Clinical trial testing.
yeah, I love those kinds of studies because they're so easy to do because the data's right there,
Exactly.
and you can just do one sitting,
This is
wait
Yeah.
for a bus. So what did you find? What kind of delays did you?
Yeah, so we found, so this is in collaboration with Patrick Ryan in Johnson. So he has a lot of a big database from his group. So he run this on his cohort on those databases. And we find a significant portion of drugs have statistically significant longer time in drug use from the EHR data than the testing time. So we have a list of drugs. And
But
then,
just generally, what's the delay look? What does it look like?
you mean
Is it 2 years,
difference?
10 years?
I should go back to that paper. I don't know. I remember it's a couple years.
Okay,
Yeah,
I'll just look it up.
yeah. It's in the paper. Sorry about that.
That's it.
Very. Yes.
This was very broad presentation. I really appreciate it. When you were talking about the big picture, next generation evidence-based medicine, the timing of adoption, you said traditionally it's post-publication guidelines, but with the next generation EPM, it's real-time decision support. Can you elaborate on that? What do you mean exactly with real-time decision support? I'm talking about like within publication and actually having that publication incorporated into EHR in real time. Is that what you mean?
Yeah, so that really refers to, the evidence-based medicine, like what policy we have, and I feel that's a gap. So for example, Stanford has this green button. I don't know if green button or blue button, which means you can just run that algorithm in a data warehouse to identify patients like this one in front of me, like for doctors, and then to look at, what's the test based on similarity metrics, you can identify what orders have been made for the patients like this one, and then generate recommendations to doctors. So we view this as kind of a real-time decision support. And then, with this large scale real world data, we can actually have a lot of new information that's not necessarily available in clinical practice guideline. But is that allowed? Are we approved to rely on these recommendations from, for real time? I think that still needs more work.
Why don't we take, there was another question in the chat.
Yes.
To them.
I think Zoom is, let's see, okay. Amy, are you still on?
Yes, although it is generally accepted that EBM is the standard of care, we see hospitals using some tools that have not been vetted. So have you seen this? And can you comment on that?
It's a standard for care and use tools which have not been vetted.
I think the question is, are you seeing hospitals use tools that are not vetted through evidence-based?
like
medicine?
FDA standards
Yeah.
or policy. Cause let's see. It's rare, I think, in Columbia, MIT, we have not allow this to happen. And also for our results, like we identify so many phenotypes. And then, you know, in web application, I showed you, you can query by patient phenotype, and you can get recommendations about possible genetic test, possible genetic disease the patient may have. We have another algorithm which is fin-to test. Like you enter a set of phenotypes of patients, we make recommendations about genetic tests that the patient should go through to facilitate the diagnosis of genetic disease. So in a research setting, they have satisfactory performance, but we have not really implemented in a care setting. I guess that needs, you know, because that affects the, how to say, really needs more validation about, you know, the safety of the algorithm and what are the possible consequences if there's an error and what's our tolerance to the error in the algorithm. So I think all this, I was, you know, my answer to the questions, I haven't seen it used very often. But Stanford does have that green button. So I think Stanford probably is a little ahead of many other institutions. So Jim, probably you know more.
I don't know about that.
Yeah.
Well, we're over time, so I wanted to be able to let people go, not feel like they're interrupting. I want to thank Dr. Weng for her presentation, and I'm sure she'll stick around a little bit to answer more questions.
Yes, I'll be happy to do that.
So, thank you. So, with other questions? Go ahead.
I have something quick. So the large language models, is there worry about the text generated by these models linking back into future training data for the future large language models, and what would be the impact of that?
You mean the output of large language model linking back to,
In the long term, yeah, to the training data of future large language models.
yeah, that could be a problem, right? Like, you know, your machine generated output become the new
Yeah.
data
New
input.
training data.
Yeah, I think that's needs more research. No, yeah, we don't have sufficient considerations for that. Yes,
I have a question. You talk a lot about the large language model, right? But how does this large language model be used to enhance our teaching?
that's a great question. Actually, I think it will be, you know, the role of large language model in education should be full of opportunities. For example, you know, the large language model can simplify the concept and enable quick question answering for students. I think it has a huge role there. Yeah. I don't expect it to be yeah.
So,
Yes.
you started talking about.
The process that is